If you spend some time exploring the forests of southern Queensland, especially the underside of leaves, you will eventually find something flashy that catches your eye. A spider with an abdomen like a disco ball. The spider genus Thwaitesia was first described in 1881 from Sri Lanka. There are currently 23 species known globally, most from Southeast Asia, but the best known are from Australia, and were fittingly discovered by a naturalist named William Rainbow. They are Thwaitesia argentio-punctata and Thwaitesia nigronodosa, the Australian mirror spiders. The spiders of the genus Thwaitesia convert their own metabolic waste into what is functionally glitter. The silver patches covering their abdomen are not made the same way as traditional arthropod coloration, as they aren't made from pigment or structural changes to the arthropod's chitin. They are crystals of guanine, a molecule that the spider's gut produced and then routed to the surface of its body via small offshoots of the digestive system, where it is stacked in plates directly beneath a transparent layer of exoskeleton. All spiders produce guanine. It is the waste product of nitrogen metabolism, the end result of protein metabolism from eating. Most animals excrete nitrogen waste through urine. Spiders, which frequently live in environments where water is rare, have solved the problem by crystallizing their nitrogen waste into solid guanine and storing it in specialized cells in the wall of their gut. In this crystalline form, guanine is chemically inert and non-toxic. It just keeps accumulating throughout the life of the spider. In most spiders, the guanine creates a flat white color, like the white patches on many common garden spiders. The guanine crystals that produce this effect are roughly cubic, scattering light in a way that makes a dull, reflective white. Thwaitesia does something structurally different. In the mirror spiders, the guanine forms thin, flat platelets stacked in layers and oriented parallel to the surface of the abdomen. The stacking produces a mirror reflection rather than random scattering. The guanine sits immediately beneath the transparent cuticle of the abdomen. In Thwaitesia argentio-punctata, these reflective deposits are arranged in discrete patches across the abdomen, intermixed but really overlapping patches of color. Thwaitesia nigronodosa forms the same patches but intermixed with dark nodes. Neither species is particularly rare in the forests of eastern Australia, but both are small enough and cryptic enough that they are routinely overlooked. So how has this beautiful but extremely impractical appearance survived? The spider covered in reflective patches sitting in a forest should be easily visible. Glitter is the opposite of camouflage. And yet Thwaitesia uses those patches as camouflage, because the forest is not a static, uniformly lit surface. It is a constantly shifting environment of dripping water, filtered light, moving leaves, and brief, direct sunlight through the canopy. In that context, a small silver point of light on the underside of a leaf is not very strange. The mirror patches are more effective camouflage in tropical areas where it rains more, because raindrops also sparkle in the sun, and so the spider goes unnoticed. This is a camouflage strategy built on matching random light events around you instead of a background color. The visual environment of a wet rainforest consists of so many points of random light and refraction, produced by so many non-spider sources, that the spider's silver patches mix in with the noise. The mirrors are camouflage because mirrors are not what predators are looking for. But how about when the camouflage fails? Thwaitesia have another behavior that is supposed to be defensive, although I don't think it is very intuitive. They have the ability to change the size of their mirrors, and in effect, change their color. When the mirrors of the abdomen reduce in size, the underlying color pattern of the abdomen becomes visible. Sometimes this can drastically change the appearance of the spider. Other spiders have exposed guanine patterns as well. Small muscles arranged under the cells containing the guanine can pull the masses together, reducing their appearance in the exoskeleton. When the muscles relax, the guanocytes spread and the silver reappears. This shift in color could be a form of startle response or a way to confuse a predator looking at the spider. The prey that the predator was about to strike at is suddenly different or harder to see. This may give the spider an opportunity to escape or evade in some way. Maybe. The function for the expanded mirrors is somewhat well known, but the function of the color pattern revealed when the silver contracts is less studied. What we do know of it comes mostly from other kinds of spiders. While there are tons of photos of these mirror spiders, relatively little is known about their specific biology.